Thank you, Donna, and thank you for the invitation. As just said, it's quite a hot topic, drug repositioning, and probably you all know what it's about. It's just a definition here from Wikipedia. Why there is a wide interest, at least from an industry perspective, is because, as you can see on that slide, you can see that the number of new drugs is pretty flat or going down, in fact, and the spend in R&D is increasing. So there's a big issue for us in terms of return on investment. And if you look at drug repositioning, what you hope is going to happen, instead of this very long, 12, 13 years time it takes, from starting a project to having something on the market, if you start, with a compound you have already, especially a compound that maybe is safe, has been through a phase 1, for example, you may reduce the time to get to market, something that's quite attractive for us. And it's because also we have lots of compounds available. So I'm just going today to tell you a few things about the kind of approaches we've been using. I'm not going to do every method we've been using. Don't have the time. I'm going to speak mainly on using genetics data. This is one of the most recent papers. I'll touch upon using expression and also side effects if I have time. Human genetics, that's why we are interested in human genetics. If you look at common disease, and you may have heard the talk by Gonzalo, the keynote talk on Sunday evening, we have all these GWAS for common disease, genome-wide association studies, and that slide you can see the growth of data. This is just a paper that has been published this month in Nature Review Genetics. And what GWAS is telling you is, if you have this is the different chromosomes here, you can see some peaks where you may have association of a disease with loci, with a genomic locus, and potentially genes, and you can zoom in and you can see more details on the different genes. As I said, there's a lot of data, so it is quite attractive because it is derived from human populations, and because there's a lot of disease being covered. This is a slide from I think December last year, and you can see different kind of therapeutic area, disease category, cardiovascular, so on and so forth. The coverage is quite impressive, across the human genome and the different category. So why we are interested in looking at genetics, especially GWAS initially for drug repositioning? There was these two papers published, one by Francis Collins in Science Translational Medicine, where he looked at different disease and found out, a lot of the hits that were derived from GWAS, were associated also with marketed drugs. The genes were associated with marketed drugs. And our colleagues and collaborators, and McGill, Brent Richards, and his collaborators published in Nature Review Genetics a specific study on OA when they found that the GWAS, again, signals, hit genes were enriched for drugs, either on the market or at least in phase 3. So that was quite interesting to us, and we thought, maybe what we can do is something much more systematic. So this is a very simple idea, in fact. And if you want more details, you can look at our paper that published in Nature Biotech a few months ago. So on one side, we take the GWAS data, and we use the NHGRI catalog, and that provides you with genes, targets, potential targets, and traits, which can be converted into disease, of course. And on the other side, we take a database that's called Pipeline, that's from pharma projects, that provides you, again, the targets of drugs that are genes, indications that are disease, and drug and compounds. And basically what we're trying to do is to bridge between these two types of data and look at drug repositioning opportunities. So of course, you can start with other type of genetics data. You can look at OMIM, for example. And on the side of the drugs, you may be able to use Drug Bank or Campbell, So here it's just a bit more details on what we've been doing. So take the GWAS genes, and I don't have the time to go to the detail. It takes quite a bit of manual curation to do that step. You take the industry pipeline, you get the genes as well, you look at common genes, and you compare the indications from the industry pipeline with the traits in the GWAS. And if the traits are similar or identical to the drug indication, so maybe that will increase the confidence in the indication. But on the other hand, when there is the trait and the disease indication are not similar, you're going to have maybe some opportunities, for drug repositioning. That's what we did. So from the pipeline, we identified 10 over 1,000 genes associated with different indication. And on that side, we identified 991 genes. And we did a filtering to get that number. Again, I have no time to go into details, but we look for replicated studies above a certain p-value. We removed traits like color of eyes or that type of things, So. And what we found was that you had 155 GWAS genes that were in common between the two, and these genes were, of course, because they are coming also from the pipeline, they were pursued by compounds and assets at different phases. So then we looked at the trait and the indication. So we found there was, increased confidence in indication for 63 genes. And of course, sometimes the gene matched to more than one disease. And for the potential drug repositioning opportunities, we had 92 genes. So if I looked at that side of the coin first, it's just a list to illustrate, some just to confirm that, GWAS is pointing to drugs that are indicated for the same indication as a GWAS trait, and that list is much longer. It's just from some example from different phases of the pipeline. Now, what was more interesting to us, in a way, was the drug repositioning opportunities when the trait was different from the disease indication. And here is just a list that give you some examples. Again, different phases of the pipeline. And you can see that there's a current drug indication, and the GWAS trait is quite different. So, and maybe those are opportunities for drug repositioning. And I'm just going to focus on the first one here, which is Denosumab, where the drug indication is OA and bone cancer, and the GWAS trait was Crohn's disease. And if you start to dig out, on information, you can find that, that make some sense, to use Denosumab for Crohn's disease. So in fact, Denosumab is an Amgen and GSK drug. There is some papers, and that Denosumab is targeting that gene, rank L here. So there's some paper, involving that gene in Crohn's and UC. What our colleagues in McGill found that the risk allele was associated with increased expression. So that was useful to know because Denosumab is an antibody. So I think he was going in the right direction, as we said. We wanted something, to reduce the expression. And we found some epidemiological data as well that was pointing that, maybe there was a link, between the OA and Crohn's disease as well. So just an example here of, when you start to connect, the dots, if you want, you can start to find, more evidence for these potential opportunities. Now, it's all very nice, but there is, of course, some challenges with using that type of data. The first one is, the gene SNP causality. So it's difficult for GWAS studies, to really, when you look at it, to really pinpoint the genes. Are you sure it is a correct gene? In fact, maybe using OMIM and we have more Mendelian disease may be easier when you know exactly what gene is involved. As I said before, the direction is a big issue as well. So you may not know if you need an activator or an inhibitor. So you may have to look, in more information or try it. So that's not always obvious. In the case of Denosumab, because we did some expression analysis on the risk allele, that was just a bit easier. And of course, if you go the wrong way, so you may create a side effects rather than a therapeutic benefit. a big issue for us was we spent a lot of time, and it was manual evaluation, manual curation of mapping the names, the traits of the GWAS and the disease indication, because there's not standardized vocabulary between these two databases. So that was quite a manual effort to do that. So to help us, what we've been looking at as well, so we've been looking at using the ENCODE data that can help you to pinpoint the gene involved, as described in a paper by Morano et al. In Science last year. Often as well, the GWAS gene may not be a good drug target. So, and we're looking, you can look also in the pathway, where that gene is, to find potentially new opportunities. And of course, we're looking at complementing that genetics approach with other methodologies we're using in the group or with collaborators in academia. Now, moving away from the genetics, we're using also expression-based approaches. And one of the most well-known approaches is called connectivity map, or CMAP in short, that was developed, in fact, 6 or 7 years ago by the Broad Institute and Justin Lamb. And this is very simple idea, in fact. So what you have on one side are the disease. You have a signature, a disease profile of genes going up and down. And what they've done is build the reference database of many compounds in a cell line. That was a breast cancer transformed cell line, MCF7. So you have that database of profile, and what you do is compare your disease profile with your reference profile. And when you have in your reference profile a signature that is the opposite of your disease profile, that may tell you that you may be able to reverse the disease by using that compound. And they had some very interesting results. So it's just one example here that came out of the lab of Atul Butt in Stanford. And there are several examples in the literature now, so, which is for IBD. So what they found is they had a negative profile for IBD with something called topiramate here. So you can see the negative profile here. So it's all very nice, but of course you want to check, and start to do some validation. And what they've done here in a mouse model of IBD, a rodent model of IBD, they've shown that by using topiramate, you can treat these mice quite well. So, and you can see the differences here as well. So they had at least some validation in a preclinical model, which is quite encouraging. So, and we've been using that kind of approach also at GSK. What we've done as well, and we published at work a few years ago, like others, you can also use geodata sets, for example, to obtain disease profile. And we've been using that as well to build disease-to-disease networks, disease-to-drug networks, so on and so forth. And you can find some unexpected links between disease and disease. What we have, and of course in the case of negative disease-drug interactions, those are repositioning opportunities. It's just an example of that big network, if you want, with tamoxifen. So you have that negative with breast cancer, not a surprise, because that's where it's been used. But you found also some interesting link with, I think one link with atopy here, one link with idiopathic myelofibrosis. You have some links with Huntington here as well. So, and you dig, if you start to look in the literature for atopy, for example, so you can start to find some evidence in models, rat models, in fact, So that maybe there is something there as well. So now, with all these expression-based approaches, again, like genetics, you have some key challenges. The quality of the disease signature, and I think we're looking very actively for more good disease signatures. So the quality, as I said, is a big issue. as I said, the initial experiment was on MCF7, so we want more cell lines to be done, maybe looking at more doses as well. So, and we've been working as well, and we published recently something on the analysis, the statistic, the methods to analyze the data. So it's something that I think we're looking actively to do. So side effects. So I just want to conclude by telling a few words about how to use side effects, because it's something we've been doing and something we published also. You can look at side effects, as therapeutic effects as something from the same kind of angle, if you want. So they may share common mechanisms, and that was our starting point. Nothing new really, because the work of Pierre Bork that was published in Science a few years ago, he looked at trying to find targets using side effects. And what we're looking is looking at therapeutic effects using side effects. And it's very, very useful or very interesting for us, because if you look at human side effects, you're really starting with human information, patient information. So you don't have that issue of translating from animal model to human. So what we've done, we took the database from Pierre Bork, which is called CIDER. On the other side, we use Pharma GKB. Basically, we mapped, you had side effects, disease, compounds here, and we mapped the disease and between the Pharma GKB and the CIDER database. So we had that mapping, and what you found is you have some drugs, so that are used to treat transplant rejection, and they are associated with side effects, CMV infections. But you have some drugs like methotrexate that is associated with CMV infection as a side effect, but not used for transplant rejection. So I think the question is then, can you potentially use that drug for transplant rejection? And in fact, when you looked at it, we found some good evidence of off-label use of methotrexate for transplant rejection. Another example here, statins, very, very famous drugs. So what we found, there's a lot of drugs that are associated, they are used to treat stroke, and one of the side effects is positive anti-nuclear antibodies. And what we found is statins, are associated with that side effect. But statins are not, at least, are not officially used, to treat stroke. And we found, again, some good evidence, in fact, from the Stroke Society that looked at various clinical trials where there's some evidence of improved results, for patients with stroke that are under statins. So we have, we've been looking, quite systematically at that type of information. And again, there's more information on our paper. So I just want to conclude by saying that, there's multiple methods that you can use with a strong computational component. And some methods are very simple. The genetics, that I described, the pipeline is extremely simple. What was very time expensive was the manual curation, in fact. There is a lot of issues when you want, to validate your findings. So that takes time. It's very difficult, to identify what the gold standards are in these type of approaches. We used a lot public domain tool, public data. We're not going to build something, unless, we really need to have to. And we try to collaborate a lot, internally and externally. So most of these projects are done in collaboration with academic groups. So I just want to acknowledge, the people that did a lot of the work. So the small team in computational biology that work on computational drug repositioning is the lead is Pankaj Agarwal. So the genetics work was done with colleagues in genetics too, and these people in CompBio. So we've been collaborating with McGill, Vincent Mousser from Lausanne, and Michael Barnes from London. So thank you.
All right. We have time for plenty of questions. If there are questions, I'd ask that if people are going to do questions, can you repeat them from the podium so they get picked up by the mic? we're waiting for people to come up with some, I actually had a couple. One of which is at the beginning when you first talked about your pipeline, you were talking about identifying whether a GWAS trait is similar to the drug indication. And there are two big questions there. One is how do you define similar, and the other is where you're getting your drug indications to. And I'm curious where those are and if you'd be better or worse by expanding them.
Okay. Shall I repeat the question still? Yeah.
Yeah, well, that one I shouted.
That's okay. So the drug indication, they came from the database called Pharma Projects that contains, the whole pipeline of the industry. So you have the drug indication. So you have the targets. The phase as well. If the project is active, not active.
Okay, so this is not just on-label use.
This is everything. Everything at different phases as well. So, and in terms the big challenge, the issue of this indication and GWAS traits, one of the biggest challenge we had to try to compare that information. And as I said, at the end of the day, it was people looking. I guess it was a human judgment at the end. To decide if it was, similar, not similar, identical, or. So
do you think you have that right, or do you think there would be some benefit to fixing that or doing it computationally?
Well, no, if there was some, but let me say that the first thing we use people that were medics, MDs, to do that work as well. Yeah. So that was useful. But if there was a way, and I'm not talking only about this indication and GWAS, but really for people to use more control vocabulary, across databases between disease, absolutely. I think, I will subscribe to that as quickly as possible, so because that's a big challenge, So when you want to cross-compare, different data sources.
Other questions, Adam? Are you using, I noticed you're talking about the connectivity map and not the links data, which is the successor to the connectivity map.
Okay. I can say a few words about that. So we are interested in links, because first, you can do more cell lines, of course, and that's the first reason. And we're looking into it. We're talking to Justin on that, and it's also because if it works, they have the new platform called L1000, where you should get, in theory, still a good profile with less genes. So basically, it'd be more cost effective to do more samples, more cell lines, more compounds, more doses that way. So,
right.
Yeah.
Right. I mean, do you feel that the data you've currently got has enough coverage of the compound space? Because that's the thing that kills me about the connectivity map. Is it's great, but I want a whole lot more drugs that they don't have.
Yeah, well, yeah. Again, there is a coverage with compound that can be better, but the coverage with cell line, I think, is key for us. So better cell lines which I feel are more relevant to disease we are interested in. So.
I'm sorry. Could you expand on that? So you think that more cell lines is more relevant than having a bigger compound?
No. I mean, compounds are both are important. So absolutely. So.
But which is more important right now?
For us, because we don't, we feel that because we done the profile of our compound collection, so we feel that adding more cell lines is more important to us.
Yeah, we have plenty of time.
Do you think it's possible to combine the current database and connectivity map data together?
So.
Is it possible to integrate the connectivity map with the current database together?
So I didn't hear the combine connectivity map with the.
With the current database about the certified or other data.
Oh, with the current database about side effects or other data? Is that right? Yeah. Which current database?
Which, yeah.
Any kind of drawback or
Yeah, well, I'm not sure exactly how you will go about doing that, So, but I think you can, because connectivity map at the end of the day, it's an expression profile, so it's different from a side effect database. But the point, there's an interesting point on side effects here. So, as I said, negative profile that points you to a drug repositioning opportunity, you know, but a positive, positive profile in a cognitive map sense, you know, may point you to potential side effects. So there's two sides of the coins in a profile, you know. So, in fact, we've been thinking about exploring more, you know, not only the drug repositioning, you know, the negative profile, but look at the positive one as well. So, which is, you know, maybe can help you to look at side effects.
Anybody else? Okay, well,
Thank
thank you
you.
very much. That's a great